So for today, my discussion would cover interaction between cells and their environments, inclusive of some information about the cell wall and of course, the overview of the endomembrane system. Okay? So the plasma membrane is actually a biological membrane that separates the interior of the cells from the outside environment, which protects the cell from its environment, which is about 5 to 10 nanometers wide. So our plasma membrane will actually have this what we call the extracellular space, and it includes your glycocalyx or the cell coat. It mediates the cell-to-cell and cell substratum interactions. It is also responsible for the mechanical protection of the cells, and it serves as barrier to particles moving toward the plasma membrane. So by checking on the image that you see on your screen right now, the glycocalyx is this part. Okay? So if this is the whole cell, this is the nucleus side, you have here your plasma membrane and then the cytosol, and outside it is the glycocalyx itself. Okay? So it is actually made of short chains of oligosaccharides. They project outward from virtually all integral proteins and some lipids in plasma membrane. And they are also closely applied to the outer surface of your plasma membrane. It also contains additional extracellular material secreted by the cell into the external space where they stay closely associated with the cell surface. So they are very important and very prominent in some types of cells like the epithelial cells lining the mammalian digestive tract. Okay? So in here, you will see figure 7.1. It shows you a diagrammatic section of the human skin, and it provides an overview of several of the subjects to be discussed in this chapter. So in here, you will see now the different adherence of the epidermal cells to another by specialized contacts. Okay? So for example, here, you will see that here how your cells are actually embedded or connected with your basement membranes, and of course how these types of adherence do appear naman between your cells. Okay, so there are attachments for the basement membrane and there are also connections between your cells. Okay, so we will be tackling most of them as we go along further in this lecture. Okay? So the extracellular matrix, or what we know as the ECM, is actually an organized network of extracellular materials found beyond the immediate vicinity of your membrane. So more than inert packing materials or non-specific glue that holds cells together, and it plays a key regulatory role in determining cell shape and the activities. So it may also consist of ill-defined amorphous associations of proteins and polysaccharides like your loose connective tissue, or may be in the form of a distinct structure. So remember that the extracellular space, of course, this is, the part wherein it is outside the cell itself. Okay, so it is the surrounding of the cell. So here, ECM takes diverse forms in different tissues and organisms, but is composed of similar proteins. And most of the proteins in these cells are compact and globular in shape, and those of the extracellular space are extended and fibrous. So among their diverse functions, the ECM proteins serve as scaffolds. So ito na naman, this is again the part or the wherein um, they serve as like the backbone or the structural support of the cell. So they are also considered as the girders. So when you say girders, these are the like if you compare it to a construction of the house, these are the metal scaffoldings or the metal supports. And then when you say mortar, this is actually like the cement that holds those connections together. And of course, it also serves like a wire, so like a binding structure. So alterations in the amino acid of the extracellular proteins can actually lead to serious disorders. So the ECM is very prominent as well in the connective tissues like your cartilages, bones, tendons, and corneal stroma. And the components of the ECM are members of the small number of molecular families like your collagens. This is one of the most important and ubiquitous ECM molecules, and they are usually fibrous glycoprotein family. Okay, so the collagen is actually the most abundant type of protein in your body. It is the major component of connective tissues and make up several body parts like your tendons, your ligaments, skin, and your muscles. Okay? And then next you have the second one would be the proteoglycans. So they are coming from the protein polysaccharide complex. So they are the protein molecules containing many bound glycosaminoglycans or the GAG chains. Okay? And then of course you have the fibronectin, laminin, and the other ECM proteins. So with the fibronectin, this is actually a multifunctional adhesive glycoprotein that plays an important role in tissue repair, in regulating also cell attachment and motility, and of course important also in embryogenesis. Okay? So for the laminin, this is the major structural glycoprotein of the basement membranes, which is which is considered to be a very intricate meshwork of proteins separating the epithelium, mesothelium, and endothelium from any connective tissue. Okay? So the basement membrane is an example of ECM and functions of the ECM proteins. So basement membrane is also known as your basal lamina. So it is a continuous 50 to 200 nanometers thick sheet. And this is actually one of the best defined examples found in the following places. So it actually surrounds your nerve fibers, muscles, and fat cells. It also underlies the basal surface of epithelial tissues such as the epidermis of the skin or the lining of the digestive and respiratory tracts. And it also underlies the inner endothelial lining of the blood vessels. Okay, so basically with the basement membrane, from the name itself, it's like if you compare it to a house, it's the flooring. Okay. So the functions of the basement membrane are the following. It provides mechanical support for the attached cells. It also generates signals that maintain cell survival. It also maintains epithelial cell polarity. It serves a substratum for cell migration and determines cell migration path. It also separates adjacent tissues within an organ, or what we all know as the compartmentalization, and it acts as barrier to passage of macromolecules and errant cancer cells. So for example, it can be very prominent in the capillary basement membranes, which actually prevent passage proteins out of the blood and into the tissues and the kidney. Okay, so the kidney is actually one of the best examples of having a very selective type of barrier when it comes to the basement membrane. So the ECM may take diverse forms in different tissues and organisms, but it tends to be composed of similar macromolecules. So unlike most proteins found inside the cell, which are very compact globular molecules, proteins of the ECM or the extracellular space are typically extended and fibrous in nature. Okay? So these proteins can self-assemble in the extracellular space, into an interconnected 3D network, and alterations in the amino acid sequence of the extracellular proteins can actually lead to serious disorders. So this image shows you an overview of the macromolecular organization of the extracellular matrix, wherein you will see here the presence of the proteins and polysaccharides. So the proteins are actually depicted by the fibronectins. Here you have the fibronectin here. You also have your collagen and of course your lamins. Okay, so they contain binding sites for one another as well as binding sites for the receptors, or what we call your integrins. So these are the considered receptor sites in the cell surface. So the proteoglycans, these smaller ones, are actually huge protein complexes that occupy much of the volume of the extracellular space. Okay? So in terms of the amount, the proteoglycan is the most abundant among them. So let's go now to the components of the extracellular matrix. So first we have again your collagen. So it comprises a family of fibrous glycoproteins that are present in the extracellular matrices, and this is found throughout the animal kingdom and are noted for their high tensile strength, having resilience to pulling forces. Okay? So kahit na kahit na medyo i-pull sila together, they have this very high resistance. Okay? But then again, of course, as people grow older, or as animals grow older, this collagen tends to, of course, degenerate. Okay? So it is also estimated that it's around 1 millimeter in diameter and can suspend a weight of about 10 kilograms or 22 pounds without breaking. So it is also very important that we also consider our weight. Usually people that do have or that are or do have heavy weights, diba? They do tend to have this friction between their collagen or these connective tissues. So nagkikis-kisan sila. So it's very important as well that we also consider the amount of weight, okay, of a certain organism. And then this is actually the single most abundant protein in the human body, having 25% of all the proteins. So collagen is produced primarily by fibroblasts, hence it is fibrous in nature, which can also be seen in other types of the connective tissues. So to date, there are 28 distinct human types of human collagen that are identified, and each type is restricted to sites in the body, but two or more can be present together in the same extracellular matrix. So functional complexity can be achieved by mixing several types in the same fiber, hence they are actually called heterotypic. Okay, so heterotypic fibers are fibers that do have two or more types of collagen mixed in together. Okay? So there are many differences among collagen family members, but they all share two important structural features. And that is, all collagen molecules are trimers consisting of three polypeptide alpha chains. So this um, these trimers can be identical or may have two or three different type of chains. Okay? So, along at least part of their length, the three polypeptide chains of a collagen molecule are wound together to form a rod-like triple helix. Okay? So, that's the confirmation of how the collagen fibers or the collagen look like. So alpha chains of a collagen molecule contain large amounts of proline And many of these proline and lysine residues are hydroxylated after the synthesis of collagen polypeptides. So the failure to hydroxylate collagen chains has actually serious consequences for the structure and function of connective tissues, like the possibility of having scurvy. Okay, so scurvy is actually a disease that results from a deficiency of vitamin C or the ascorbic acid and is characterized by inflamed gums and tooth loss, poor wound healing, brittle bones, and the weakening of the lining of blood vessels causing internal bleeding. Okay, so there are also fibrillar collagens. They are, or there are three types. You have type 1, type 2, and type 3. They assemble into rigid cable-like fibrils, which in turn assemble into a thicker fiber that are typically large enough to be seen under the light microscope. Okay? So fibrils are strengthened by covalent cross-links between lysine and hydroxylysine residues on adjacent collagen molecules. So if they are disrupted or if they are broken, it will be weakened, which will result in decreased elasticity, such as what you can observe in your skin. So, and it can also actually increase brittleness of the bones among the elderly. Okay? So the structure or the image that you see on your screen right now is actually the structure of your collagen. Okay? So the collagen provides insoluble framework that determines many of the ECM mechanical properties and 3D organization of collagen molecules that often correlates with the properties of tissue which it is found. So for example, in the tendons, so these tendons do connect muscles to bones. So they must resist large pulling forces during muscle contraction. So the role of the collagen fibrils are aligned in parallel to long axis of the tendon and parallel to the direction of the pulling forces. In the cornea, it serves as a durable, transparent protective layer at the eyeball surface so that actually, or the light can pass through easily. And in the basement membrane, collagen are very thin and they are considered to be as mechanically supported sheets. They are organized into network that provides mechanical support and serves as a lattice for the deposition of other extracellular materials. So, given their abundance and widespread distribution, serious disorders can be caused by the abnormalities in the fibrillar collagen formation. So, for example, if you experience to have burns or traumatic injuries to the internal organs, it can cause scar tissue buildup. So largely fibrillar collagen are responsible when it comes to wound healing. Okay, but if you will be having, an abnormal amount of fibrillar collagen in your body, of course, wound healing wouldn't be so easily, be started in your body. Okay? So because these fibrillar networks are the ones patching up the wound or to close up the wounds that can be a result of having burns or traumatic injuries. Okay? So for type 1 collagen mutations, you can have osteogenesis imperfecta. This is potentially lethal condition characterized by extremely fragile bones, thin skin, and weak tendons. And then next we have the type 2 collagen mutations. It can alter cartilage properties and causes dwarfism and skeletal deformities. Okay, so in here you will see that with dwarfism, of course, the growth of the bones are not normal. Okay, and then a number of collagen gene mutations which cause various distinct but related collagen matrix structure defects, such as your Ehlers-Danlos syndrome, which one causes hyperextendable joints and highly extensible skin. And then you also have the type 4 collagen gene mutation like the Alport syndrome. It is an inherited kidney disease in which the glomerular basement membrane is disrupted. Okay, so it's very important in the glomerular basement that it is, that it does have this um, filter support, because as you know, the glomerular basement are found in your kidneys. So if these membranes are usually disrupted, of course, uh, there's no um, there's no use that your kidney would be able to filter out the waste coming in and out of the body, and of course the regulation of the salt. Okay. So in addition to collagen, basement membranes and other extracellular matrices typically contain large amounts of distinctive type of protein, protein polysaccharide complex known as your proteoglycan. So it consists of a core protein, which is again the glycosaminoglycan chains, or the GAG chains, that are covalently attached. So the GAGs are actually composed of repeating disaccharide having a structure of ABAB, where AB is actually a representation of two different sugars. So GAGs are very acidic due to both carboxyl and sulfate groups on their component sugar rings, and due to the sulfated GAG negative charges, proteoglycans bind large numbers of cations, which in turn attract lots of water molecules. So here you will see how the structure of a cartilage type proteoglycan complex look like. So here you will see the what you call the core protein that are attached to a single proteoglycan. Okay, so so you will see here that it can be or the core protein can be linked into chondroitin sulfate, keratan sulfate, and of course hyaluronic acid. So in addition to serving as major ECM component, one group of the proteoglycans, the heparan sulfate proteoglycans, or what you can abbreviate as HSPGs, function directly at the cell surface. So members of this group of proteoglycans include the syndecans, whose core protein spans the plasma membrane, and the gags of these proteoglycans are not constructed of identical disaccharides, but instead exhibit structurally diverse disaccharides. So next, let's go to the other components, including the fibronectin, laminin, and other ECM proteins. So the matrix implies a structure made up of a network of interacting components. This is up for the ECM. So it contains a number of proteins in addition to collagen and proteoglycans that interact with one another in highly specific ways. So many of the ECM proteins occurs in families, and more than one is formed, each formed by alternate mRNA splicing. So first, let's go to the fibronectin, or the fibrous type of extracellular component. So this is actually one of the best studied ECM proteins. It has features common to most other matrix components and ECM proteins. It consists of linear array of distinct building blocks, which is in a modular construction, and each fibronectin polypeptide is constructed from a sequence of around 30 independently folding FN modules of three distinct types. So you have FN1, FN2, and FN 3. So each of the two polypeptide chains making up fibronectin contains first the binding sites for the ECM components like your collagen and proteoglycans, and this links the molecule into stable interconnected networks. So the binding sites for cell receptors, they form stable ECM cell attachments, and the endothelial cell will adopt shape unlike it does in the body when it spreads over a square surface coated with fibronectin. So the importance of the fibronectin and other ECM proteins is particularly evident during the embryonic development. So for example, you have here the neural crest cells following fibronectin pathways out of the developing nervous system into virtually all the parts of the embryo, and they traverse pathways in rich in fibrils composed of the fibronectin. So anti-fibronectin antibodies disrupt other developmental processes, like for example, in the mice lacking functional fibronectin gene would result to an abnormal mice which actually do not survive past early development. So for the fibronectin structure, please check on page 230 of your book, okay, to see how this fibronectin structure look like. Next, laminin. This is also one of the specific type of protein in your extracellular matrix. So this came from a family of extracellular glycoprotein, which consists of 3 different polypeptide chains linked by disulfide bonds. It is organized into a cross with 3 short arms and 1 long arm. So it is extracellularly found. It greatly influences cells' potential for migration, growth, and differentiation. So certain cells migrate over laminin-containing matrix that they secrete some substances as well, like in your keratinocytes. They also bind tightly to other laminins, proteoglycans, basal lamina components, and cell surface receptors. So in fact, basement membranes fail to form in mouse embryos that are unable to synthesize laminin, causing the death of the embryo around the time of implantation. So the ECM can also exhibit dynamic properties both in space and over time. And ECM degradation along with that of the cell surface proteins is accomplished by largely by a zinc-containing enzyme family or for example, your matrix metalloproteinases or the MMPs. Okay? So other ECM molecules includes your tenacin. These are large oligomeric glycoprotein found on many cell types and a variety of cancer cell surfaces depending on cell type, which promotes or discourages cell adhesion. Next, you have the Antactine. This is a component of the basement membranes. It is thought to play a role in adhesion and penetration of early mammalian embryo into the uterine lining. And then you have the thrombospondin, which is secreted into the ECM by many cells. And this is prominent in the matrix surrounding the lining of mature blood vessels and inhibits formation of new blood vessels like in your angiogenesis. So usually, angiogenesis is found in the formation of chick embryos. Okay, so usually the growth of the blood vessels in the chick embryo is within the process of angiogenesis compared to humans which use vasculogenesis. Okay, so in angiogenesis, this is actually or it has the idea of this vascular vessels arising from each other compared to the vasculogenesis. Okay. Next, you have the interactions among ECM materials that can be very complex. So, some many containing binding sites with opposite activities. So, one may promote and the other can inhibit cell adhesion. It does affect one cell behavior in many and can have many changes from time to time. So there are also spatial arrangements of various components that may ultimately determine their effect on cell behavior. And ECM components like fibronectin, laminin, proteoglycans, and collagen are capable of binding to receptors situated on the cell surface. So next we have the integrins. So for the confirmation of or how the integrins look like, you may want to open or check your book on page 232. So integrins are integral membrane protein family. They are composed of two membrane spanning chains, which includes your alpha and beta linked non-covalent bonds. So they are thought to be on the surface of virtually all vertebrate cell types and found only in animals. So this is the most important family of receptors that attach cells to their extracellular environment, and they bind to specific substances in the extracellular environment, or the ligands. So there are 18 different alpha and beta subunits which are identified on the cell surfaces, and thus many heterodimers, and each with specific distribution within the body. So there are only around 24 integrins identified on the cell surfaces. So um, for EM pictures or extra or for the EM pictures in the late 1980s, they suggest that two subunits are oriented to form a globular extracellular head connected to the membrane by a pair of elongated legs. So each integrin can bind a number of divalent cations like your calcium, magnesium, and manganese, although the effect of each ion on the structure and ligand binding capabilities of the proteins remain unclear. So if you see when you say cations, these are positively charged molecules or ions. Okay? So in 2001, the first X-ray crystallographic structure of an integrin extracellular portion is displayed with a highly unexpected feature. So many integrins can exist on surface of cell in an inactive conformation. They can be activated rapidly by events within the cell that alter the conformation of cytoplasmic domains of the integrins subunits. So the changes are propagated through the molecule, increasing the integrin's affinity for extracellular ligand. For example, the platelet aggregation during blood clotting occurs only after cytoplasmic activation of A, 2B, B3 integrins, which increases their affinity for fibrinogen. Okay? So when it comes to the when it comes to wound healing, there are actually different components that are responsible for it. So one type of this components come after the other. Okay, so kumbaga they have this order. Hindi po pwedeng mauna yung isa kaysa dun sa isa. Kailangan meron munang mga mauunang substances dun na pupunta. Kumbaga, sila yung mga nauunang um, mag-rescue dun sa site of infection or to stop the infection in wounds. Okay? Bago magkaroon or bago pumasok yung ibang extracellular membrane components to patch up the wound itself. Okay? So evidence suggests strongly that the bent conformation of an integrin corresponds to the inactive state and cannot bind a ligand. And studies suggest that ligand binding ability of integrin head projecting from the plasma membrane outer surface depends upon special arrangement of the alpha and beta cytoplasmic tails present on the membrane inner side. So integrins are implicated in two major types of activities. The first one would be the addition of cells to their substratum or to other cells. And the second one would be the transmission of signals from external environment to the cell interior or outside in signaling. So outside-in signals transmitted by integrins and other cell surface molecules can influence many aspects of cell behavior, including differentiation, motility, growth, and even cell survival. So influence of integrins on the cell survival is best illustrated by comparing normal and malignant cells. Linkage between integrins and their ligands mediates adhesions between cells and their environment. And most extracellular proteins that bind integrins have tripeptide sequence, which includes the arginine, glycine, and aspartic acid. So applications and examples of integrin involvements are linked to the discovery of the RGD sequences, which give importance that have opened the door to new treatments of medical conditions involving receptor-ligand interactions. So when the wall of a blood vessel is injured, the damaged region is sealed by the controlled aggregation of blood platelets, which are non-nucleated cells that circulate the blood, and the integrin cytoplasmic domains contain binding sites for a variety of cytoplasmic proteins, including several that act as adapters to link the integrin to actin filaments of the cytoskeleton. Okay, so that's how the integrins actually work with other cytoplasmic proteins to be able to close wounds. Okay. So in here, you will see the Integrin Confirmation, which is figure 7.3 in your book. So that's actually how it looks like. This is the alpha and the beta conformations and how they are, working with the different types of cytoplasmic components. Okay, so in here you will see that it is with the attachment of tallinn. So when this tallinn is attached to one of the ends of this integrin conformation, it gives the signal to have these integrins be attached to different cytoplasmic components like your collagen. Okay, so also it has a role in the platelet aggregation. So in here, you will see that there, for example, in the site of the injury, you have the platelet receptors, and from there, the platelet adhesion happens, and then it also binds, or the integrin has a role of this, the role of, connecting another platelet to another platelet. Okay, so with this one, of course, it would also produce, what they call the RGD peptides. Okay, so with the presence of the RGD peptides, it can actually inhibit blood clot formation by competing with the fibrinogen molecules for the RGD binding sites. Okay, so clearly, if the RGD peptides are present, it will inhibit blood clot formation. This is actually one of the things that we should, expect when you have wounds, right? That there should be a blood clot to stop the blood from going out of the wound. Okay, so if there's the presence of more of these integrins, of course it would attract more of the platelets, okay, going into the site of injury. But if there are RGD peptides, these peptides would actually inhibit the um, blood clot formation. Okay? Next, we have the focal adhesions and hemidesmosomes. So this now will be the interactions of the cells with non-cellular substrates. So focal contacts anchor cells to their substratum. It is much easier to study cell adhesion to a surface in vitro or in cell culture dish than with extracellular matrix inside an animal. So much of our knowledge of the cell matrix interactions is derived from studies of cells adhering to various substrates in vitro. So focal adhesions are actually dynamic structures, and they can be rapidly disassembled if the adherent cell is stimulated to move or enter mitosis. So focal adhesions have been implicated in cell adhesion and or cell locomotion, although the precise roles of these structures have been debated over years. Conversely, mechanical forces applied to cell surfaces can be converted by focal adhesions into cytoplasmic signals. So here you will see the figure showing the focal adhesions, which are the sites where cells adhere to their substratum and transmit signals in both directions across the plasma membrane. So in here, you will see in Image A, this is actually a cultured cell that has been stained with fluorescent antibodies that reveals the location of the actin filaments. So the actin filaments are actually the gray-green in color, and then the red ones are the integrins. And the integrins are localized in small patches that corresponds to the site of focal adhesions. And then for image B, this one, this is the cytoplasmic surface of the focal adhesion of a cultured amphibian cell, which is showing the inner surface of the membrane that was processed for quick freeze deep etch analysis. So bundles of the actin filaments are seen to associate with the inner surface of the membrane in the region of the focal adhesion. And then figure C, we have here a schematic drawing of the focal adhesion showing the interactions of the integrin molecules with other proteins on both sides of the lipid bilayer. So this is how, the integrins would actually or would have um, shows how the focal adhesions work with the other parts of the extracellular matrix. So here you have the ECM, you have the integrins, and then the plasma membrane. So outside the plasma membrane, you will see the integrin signaling layer, and then you have the force transduction layer here, and then here the actin regulatory layer, and then the actin cytoskeleton. So again, basically, they do have these layers, no? They don't really mix up with each other. So focal adhesions are most commonly seen in cells grown in vitro, but similar types of adhesive contacts are found in certain tissues like the muscles and the tendons. So in the body, the tightest attachment between a cell and the ECM is at the epithelial cell basal surface, where they are anchored to underlying basement membrane by specialized adhesive structure called your hemidesmosome. So they contain dense plaque on membrane inner surface with filaments coursing outward into the cytoplasm, and bullous pemphigoid is actually a rare autoimmune disease that demonstrate importance of the hemidesmosomes. So this is actually a skin condition that causes large fluid-filled blisters, and it is because people make antibodies against hemidesmosome plaque proteins. So these are the bullous pemphigoid antigens. Okay, so if there are these antibodies, this is actually what happens to those people that do have this bullous pemphigoid autoimmune disease. Okay, so instead of you having a very, elastic type of skin, what happens is that, of course, you will be having this fluid-filled blisters on your skin. Okay? So when you say an autoimmune disease, this is also directed by the immune system. Okay, so in here, you will see the images of the experimental demonstration of the forces exerted by focal adhesions. And then here you will see how the hemidesmosomes look like. So they are differentiated in sites at the basal surface of the epithelial cells, where the cells are attached to the underlying basement membrane. Okay? So the experimental approaches, this little is known about the mechanisms that generate complex 3D cell arrangements in developing organs. So it is presumed that it actually depends heavily on selective interactions between cells of the same and different types. So experiments demonstrating cell adhesion are difficult to study, occurring in small organs of a developing embryo. So there are four distinct families of integral membrane proteins that play a major role in mediating cell-to-cell adhesion. So first we have the selectins. Second will be members of the immunoglobulin superfamily or IGSF. We also have members of the integrin family and cadherins. So selectins comprise an integral membrane glycoprotein family that binds to specific sugar arrangement in oligosaccharides that project from other cell surfaces. So the name of this class of cell surface receptors comes from lectin, a term for a compound that binds to specific carbohydrate groups. So during the 1960s, they have removed lymphocytes from peripheral lymph nodes and labeled them radioactively and re-injected. So they go back to the site of the origin from which they were derived, which is actually called the lymphocyte homing. So selectins possess a small cytoplasmic domain, a single membrane spanning domain, and a large extracellular segment consisting of a number of separate modules, which is the outermost domain that acts as the lectin. So there are also three known types. You have the E-selectin, which are usually found in the endothelial cells, P-selectin, found in your platelets and endothelial cells, and then L-selectin, which are present on all types of leukocytes or white blood cells. Next, you have the immunoglobulins or IGs and integrins. They are blood-borne antibodies or the immunoglobulins, and their structure was elucidated in the 1960s, and this was actually a milestone in understanding of the immune response. So IGs or the immunoglobulins are large family of proteins, and they consist of polypeptide chains composed of a number of similar domains, and most are present on lymphocyte surfaces as integral proteins. So most of the um, immunoglobulins or the members of it are involved in various aspects of immune function, but some of them mediate calcium-independent cell-to-cell adhesion. And most immunoglobulins mediate specific interactions of lymphocytes with cells needed for the immune response. For example, like targeting lymphocytes, macrophages, and target cells, but some mediate adhesion between non-immune cells. So if you remember, we have the different types of immunoglobulins like your IgG, IgA, IgM, IgG, and IgE. Okay, so those are the different types of your immunoglobulins, and they do have this specific, function that actually combats some infections in your immune system. Okay, so like fibronectin and other cell adhesion proteins, immunoglobulins self-adhesion molecules exhibit modular construction. They are composed of individual domains similar in the structure of the domains in other proteins. And then L1 importance is actually important in neural development and has been revealed in several ways. Like human L1 gene mutations can have devastating consequences. So various types of proteins serves as ligands for immunoglobulin cell surface molecules and may bind to same or different immunoglobulins in the other cells or a few integrins. Next, we have the cadherins. This is a large family of glycoproteins mediating calcium-dependent cell-to-cell adhesion. Also, they transmit signals from the ECM to the cytoplasm and found in many different cell types with specific body distribution. So typically, they join cells of similar type to one another, mostly by binding the same cadherin present on the neighboring cell surface. This was demonstrated by the following experiment. So like selectins and immunoglobulin molecules, cadherins have a modular construction, and the classical cadherins have a relatively large extracellular segment consisting of 5 tandem domains of similar size and structure, and one transmembrane segment and a small cytoplasmic domain. And the X-ray crystallography has been carried out on the extracellular portions of the cadherins. So, cell-to-cell adhesion results from the interaction between the extracellular domains of the cadherins from opposing cells form a cell adhesions zipper. So, which, if extensive, would hold cells together with great strength. Cadherin-mediated adhesions may be responsible for the ability of like cells to sort out of mixed aggregates, and then mesodermal cell movement is in gastrulation exemplify mesenchymal epithelial transition. And cadherins and other cell adhesion molecules play a key role in such activities by changing cell adhesive properties. Okay, so basically these cadherins would actually can actually provide also great linkage from one cell to another, but of course with the presence of what you call your calcium ions. Okay? So here you will see the overview of the cell adhesion molecules, including the selectins, how do they look like. You also have the L1 proteins and how do they interact with the other immunoglobulin domains. And then you also have your integrin and immunoglobulin domain interactions. And these are the cadherins with the presence of your um, calcium ions. Okay, so that's how you differentiate one from the other. So adherence junctions and desmosomes. So these are the other anchoring cell this is actually, or these are the ones responsible for anchoring cells to other cells. Okay, so cells of certain tissues like your epithelia and cardiac muscles are notoriously difficult to separate from one another because they are held together tightly by specialized calcium dependent adhesive junctions. So these are actually what we call your adherence junctions, and they are found in variety of body sites, particularly common in the epithelia. Okay? So, they occur as belt circling each cell at apical end and binds cell to its surrounding neighbors. They may also transit signals between them, which is what you call disonule adherence. Okay? So thus junction cadherents like focal adhesion integrins connect the external environment to actin cytoskeleton and provide a pathway for signal transmission from cell exterior to the cytoplasm. So this is a schematic diagram of an intercellular junction complex. So this is now for you to be able to further understand how these different all of the information that we have discussed earlier works in your extracellular matrix. Okay, so if this is the extracellular matrix, how are your cells linked to the extracellular matrix? So you have here your hemidesmosomes. So what links your cells from another cells? You have what we call your desmosomes and intermediate filaments. You also have the adherence junctions. You also have the gap junctions, wherein this is the site wherein the signals are being transferred from one cell to another, and also you have the tight junctions. Okay. So desmosomes, or what you also know as the maculae adherens, is a disc-shaped adhesive junction which is around 1 micrometer in diameter and are found in a variety of tissues. So again, most notably in your epithelia, and where your basal, um, where they are basal to the zonulae adherents. So particularly numerous in tissues subjected to mechanical stress, like your skin epithelial layers, cardiac muscle, gingiva or the gums, and the uterine cervix epithelial layers. So like adherence junctions, they contain cadherins that link the two cells across a narrow extracellular gap. They are dense cytoplasmic plaques on the inner membrane surfaces that serve as sites of anchorage for looping intermediate filaments. And the region between cells, what we call your desmoglea, are filled with tightly or lightly staining materials which may act as glue. And autoimmune disease, pemphigus vulgaris, illustrates importance of cadherins in maintaining epithelium structural integrity. So the role of cell adhesion receptors in the transmembrane signaling. So all four types of cell adhesion molecules have the potential to transfer information across the plasma membrane. So the integrins and cadherins can transmit signals from extracellular environment to the cytoplasma via links with cytoskeleton and with cytosolic regulatory molecules like your protein kinases and G proteins. So the engagement of an integrin with its ligand can induce a variety of cell responses via transmembrane signaling, and such changes can alter subsequent cell behavior like your growth potential, migratory activity, differentiation state, survival, and an example would be the mammary gland epithelial cells. So remove cells from the mammary gland and you could actually grow them on bare culture dish. In absence of the extracellular glycoproteins laminin and fibronectin, they appear as flattened and undifferentiated cells and lose their ability to produce milk proteins. So if you add laminin or other extracellular molecules to the culture, it binds cell surface integrins, and then in return, cells regain their differentiated appearance. So laminin may work by binding cell surface integrins and activating kinases at the inner membrane surface. Next, we have the tight junctions. This is sealing the extracellular space. So it is a simple epithelium is comprised of a layer of single cells that adhere tightly to one another to form a thin cellular sheath. So the tight junctions, or what we call your TJs or the zonula occludens, has been discovered in the 1960s, and they are found between adjacent epithelial cells at the very apical end of the junctional complex between those cells. So in tight junctions, the membranes can contact at the intermittent points, not fused over large surface areas. The points of the cell-to-cell contacts are sites where integral proteins of the two adjacent membranes meet within the extracellular surface. Thus, your tight junction serves as a barrier to free diffusion of water and solutes from extracellular compartment on one side of epithelial sheet to that on the other side. So TJs also serve as fences that help maintain the polarized character of epithelial cells. Thus, they block diffusion of integral proteins between the apical domain of the membrane and its lateral basal domains. So TJs are occluding junctions and form a continuous permeability barrier, but not all of them exhibit the same permeability proteins. So TJs with several parallel strands form better seals than those with one or a couple of strands. Some tissues are permeable to specific ions or solutes to which other TJs are impermeable. Not until 1998, TJ strands were thought to be composed of single protein, what you call the occluding proteins. And the example of permeability differences is seen in one small region of the human kidney tubule, specifically the thick ascending limb or the tal. And the tal has tight junctions that are permeable to magnesium ions. So another important function that came to light in 2002 about your tight junctions is that for decades, mammalian skin impermeability to water was solely thought to be a property of the outer cornified layer of the skin, which contains tightly packed protein filaments and associated lipids. So Chukita et al. In 2002 discovered, however, that mice lacking a gene for Claudin-1 died shortly after the birth as a result of dehydration. Okay, so there's an importance of this Claudin-1 when it comes to the permeability of water in the skin. Okay, so tight junctions also present between endothelial cell lining capillary walls are particularly evident in the brain as well. So they help form the blood-brain barrier, which actually prevents substances from passing from bloodstream into the brain. Although small ions and even water molecules may not be able to penetrate the blood-brain barrier, immune system cells are able to pass across the endothelium through these junctions. And while protecting the brain from unwanted solutes, the blood-brain barrier also prevents access of many drugs to the central nervous system. So this one would actually, or these images would show you how the different or how the tight junctions look like under the microscope, and of course a schematic diagram. We have here the electron micrograph, okay, of a particular tight junction, and this is actually the schematic diagram. So you have here the tight junctions in between your plasma membranes. Okay. Next, let's go to the gap junctions. These are the mediating intercellular communication, or these are the ones responsible for the intercellular communication. So, gap junctions are found in the plasma membranes of adjacent cells that come very close together, which is around 3 nanometers in diameter, but they make no direct contact. They are sites between the animal cells that are specialized for intercellular communication, and they are very fine strands that span gap between cells and form molecular pipeline connecting adjacent cell cytoplasms. So, link cells of most mammalian tissues except skeletal muscle and most nerve tissues. So, gap junctions have simple molecular composition made entirely of integral membrane protein connexin, which actually came from a multi-gene family, and there are around 12 types of connexin that has been reported. So, connexins clustered in membrane to form multi-subunit complex you call your connexon, and totally spans the membrane. So during gap junction formation, connexons in membranes of opposing cells become tightly linked to one another through extensive interactions of the connexin subunit extracellular domains. So gap junctions mediate the gap junction intracellular communication, or what we call the GJIC. These are communication sites between the adjacent cell cytoplasm. So this gap junction intercellular communication is revealed through passage of ionic currents or low molecular weight dyes using the fluorescein from one cell to its neighbors. So gap junctions are also thought to be gated. So channels or channel closure is probably triggered primarily by phosphorylation of connexin subunits by protein kinases. So while skeletal muscle cells are stimulated by chemicals released from the tips of nearby nerve cells, cardiac or smooth muscle stimulation occurs by a very different process, one involving your gap junctions. So for example, contraction of mammalian heart is stimulated by an electrical impulse generated in the small region of the specialized heart muscle sinoatrial node, which acts as the heart's pacemaker. And gap junction also occur between the presynaptic and postsynaptic membranes of adjacent nerve cells in certain parts of the brain. Next, gap junctions can put many cells of tissue into intimate cytoplasmic contact and has important physiological consequences. This is with respect to the molecules that are small enough to pass and connected cells to form one giant compartment. So highly regulatory or highly active regulatory molecules, or CAMP, specifically inositol phosphates, pass through gap junction channels. So connected cells respond together even if only a small portion of the cells are stimulated by the hormone. And gap junctions allow cells to cooperate metabolically by sharing key metabolites, for example, inclusive of your ATPs, sugar, phosphates, amino acids, and many coenzymes, which are small enough to pass through these intercellular channels. So connexins are the proteins of which gap junctions are made and are members of a multi-gene family. So again, around 20 different connexins with distinct tissue-specific distributions have been identified, and these compatibility differences may play important roles in either promoting or preventing communication between different types of cells in an organ. So here you will see how the gap junctions look like under an electron micrograph, and of course a schematic diagram. So these are the gap junction particles, or the connexons, found in your outer side of your, or in the surfaces of your lipid bilayer. And then you have here and it does have these hydrophilic channels. And then, of course, the connexon has or is composed of connexin subunits. Okay, so this gap junctions would actually remind you of the flour donuts, if you're familiar with them, but they look like those. Okay. So next we have the Plasmodes Mata. So the Plasmodes Mata is like gap junctions of animal cells. They are cylindrical cytoplasmic channels which are around 30 to 60 nanometers in diameter that pass through the cell walls of adjacent cells. And plasmodesma is actually the singular term for it. Okay? So they are aligned by plasma membranes and usually contain a dense central structure, or what you call the desmotubule. Okay? So they serve as sites of intercellular communication, allowing plant tissue to function as metabolic unit. And the desmotubule is derived from smooth endoplasmic reticulum of two cells, which is the passage usually limited to space between desmotubule and the inner membrane surface. So unlike gap junctions, which have fixed openings, the plasmodesmatal pore is capable of dilation, so larger molecules can pass, like your RNA and your proteins. So It was shown later that plant cells make their own movement proteins that regulate the flow of proteins and RNAs from cell to cell. So this is the schematic diagram of a Plasmodes Mata. So the desmotubule is here, found at the center. So this part of the Plasmodes Mata is very flexible to allow larger molecules to pass through it. Okay. So next, let's move on now to the cell wall. Okay. So in the cell walls, so they have this what we call the naked cells, which are covered by a more than um, 10 nanometers lipid protein membrane that are extremely fragile structures. So this offers minimal protection for cells, and cells of nearly all organisms but animals are enclosed in a protective outer envelope. Okay? So the vital functions of the plant cell walls um, develop osmotic turgor pressure that pushes against their surrounding wall, and the wall gives the enclosed cell its characteristic polyhedral shape. Okay? So in addition to providing support, cell walls also serve collectively as type of skeleton for the entire plant. So cell walls also protect the cell against damage from mechanical abrasion and pathogens, and they also mediate cell-to-cell interactions. So like the ECM at the animal cell surface, plant cell wall can also be a source of signals that alter the activities of the cell that it contacts. And this is also a primary barrier to large molecular substance penetration, while ions or small molecules could actually pass freely. So compared to your animal cell, the cell walls are more rigid compared to them. So this is why it explains um, why the animal cell actually has a similar size all throughout its development. So the differences would just happen only after fertilization. But in plant cells, cells do actually grow in a manner of elongation and the diameter expansion. Okay. So, kumbaga, the cells within, the cell walls would actually just develop not in, they are not like the animal cells that do actually divide symmetrically. Okay? So, in the cell walls or in plants, cells do actually divide asymmetrically. It can be in an elongated path or in an elongated direction or in a cross-sectional or in a lateral type of growth. Okay? Hindi pataas, pwedeng palapad. Okay? So the cell wall structure is like to fabricated materials like reinforced concrete or fiberglass and contain fibrous element embedded in non-fibrous gel-like matrix. So cellulose is the fibrous component of the cell wall, and it is organized into microfibrils that confer rigidity on cell wall and provide resistance to tensile or pulling forces. So proteins and pectin provide the matrix. So cellulose molecules are polymerized at the cell surface, and glucose subunits are added to the end of growing cellulose molecules by multi-subunit enzyme complex embedded in the membrane, what we call your cellulose synthase. So the cell wall matrix is made up of three types of macromolecules. So you have first your hemicelluloses, which are branched polysaccharides whose backbone is consisted of one sugar like glucose and side chains of other sugars like your xylose. And hemicellulose molecules bind to cellulose microfibril surfaces, cross-linking them into a complex and resilient structural network. And then next, we have your pectins. These are heterogeneous class of negatively charged polysaccharides containing galacturonic acid. And then proteins, which are the functions of this are well not understood because proteins are usually found in your diba, in your animal cells, but it is less likely that we find proteins in plants. Okay, but they actually mediate dynamic activities. Okay, if animal cells do have this huge amount of proteins made that actually makes it for plant cell walls, their abundance is very very minimal in number. So percentages of these various materials in the cell walls are highly variable. So it actually depends on the type of plant and type of cell and stage of the wall. So cell walls are dynamically structures modified in response to the changing environmental conditions like animal connective tissues in the ECMs. So this picture that you will see here is actually the plant cell wall, showing you the position of the hemicelluloses. Those are the ones that are, in violet in color. You also have the pectin, which is the one in blue color. And then you have the strands of glycoproteins, the ones colored in brown. So compared to the number of the pectin and hemicelluloses, and of course the other um, microfibrils, the glycoproteins are less in number. So cell walls arise as a thin cell plate formed between membranes of newly formed daughter cells after each cell division. So cell wall matures by incorporating additional materials assembled inside the cell and then secreted into the extracellular space. And walls of young, undifferentiated cells must be able to grow along with the enormous growth of cell it surrounds, along with providing mechanical support and protection from foreign agents. And the transformation from primary to secondary wall occurs as wall increases in cellulose content and in most cases incorporates lignin. So lignin is a phenol-containing polymer that provides structural support. So next, let's go to the endomembrane system, or the overview of the endomembrane system and its dynamic nature. So before the 20th century, experiments actually performed were by staining tissue sections hinted at an extensive membrane network in the cytoplasm. So in the 1940s, the endomembrane is revealed as diverse array of membranous structures in the cytoplasm of most eukaryotes, and these studies and subsequent biochemical studies showed that eukaryotic cell cytoplasm was subdivided into a variety of distinct membrane-bound compartments. And mitochondria and chloroplast are not part of this interconnected system. So current evidence suggests that peroxisomes have a dual origin. So the image that you see right now is the membrane-bound compartments of the cytoplasm, showing you the Golgi complex, endoplasmic reticulum, and of course your excretory vesicle. So these organelles are part of the dynamic integrated network, and materials are shuttled between parts of the cell. So transport vesicles shuttle things between organelles and form by budding from donor compartments. So when you say budding, they just separate from from the parent. They just separate and then grow to a uh, new and same form of organelle. So transport vesicles move in directed manner, often pulled by motor proteins operating on tracks formed by microtubules and microfilaments of the cytoskeleton. So when they reach their destination, they fuse with acceptor compartment, which receives vesicles, soluble cargo, and membrane wrapper. So they exhibit repeated cycles of budding and fusion that move a diverse array of materials along the numerous pathways traversing the cell. And several distinct pathways through the cytoplasm have been identified and they fall into two groups. The first one would be a biosynthetic pathway, and the second one is an endocytic pathway. So the biosynthetic pathway or the secretory pathway includes the synthesis in the endoplasmic reticulum of proteins or Golgi apparatus for the lipid and carbohydrate synthesis that are altered as pass through the Golgi sent from their various locations in the membrane, including the lysosome, large plant cell, vacuoles, etc. So many materials made in the ER and Golgi are fated for secretion from the cell. And two types of secretory activities are actually present. The first one would be the constitutive, and the other one is the regulated secretory activity. So proteins, lipids, and complex polysaccharides are transported through cell along biosynthetic or secretory pathway, and discussion will center on several distinct classes of proteins. So next we have the endocytic pathway. So it moves materials or membrane surface into a cell from outside to cytoplasmic compartments like the inclusion of your endosomes and lysosomes. And movement direction is opposite to that of the secretory pathway. Okay? So hence, when you say secretory, it should be going out, and then endo, it means going in. Okay? So this is an overview of the biosynthetic and secretory and endocytic pathways that unite the endomembranes into a dynamic interconnected network. So just study or read up on this image. Okay? So you could actually follow how the processes happen. So next, proteins targeted to specific destinations through sorting signals located on them and receptors in transport vesicle walls that recognize them. So you could do this by having the analogy of trucks carrying different cargo to various sites. So both transport pathways require defined traffic patterns to ensure accurate delivery to the correct sites. So targeting involves integral membrane proteins, secretory proteins, and lysosomal proteins, which are routed to their appropriate cellular destination by the virtue of specific addresses or sorting signals. So sorting is facilitated by specific membrane receptors for sorting signals found in particular membranes of the endomembrane systems, or by coats that form an outer surface of the transport vesicles. So emission micrographs give detailed view of the cell cytoplasm but little insight into functions of the structures. So insights gained from the autoradiography actually detected the location of the or can actually detect the location of a radioactively labeled materials in the cell. So, for example, we have here the pancreas assigner cells that have a particularly extensive endomembrane system, which is very ideal for the study of autoradiography. So James Jamieson and George Palad worked with the pancreas assigner cells, followed secretory proteins from the synthesis to secretion, and determined individual steps even though all of them occurred simultaneously. So the procedure, they sectioned tissues containing radiolabels, and they located hot digestive enzymes with autoradiography. So the insights from the pulse chase trials show secretion proteins path after synthesis to their site of discharge. So exposed to hot amino acids briefly pulse, followed by a wash to remove excess isotope from the tissue. So transferred tissue to the medium with unlabeled amino acids or the chase actually lasted for varying time periods. So one can see wave of radioactivity moving through the cell, and they discern this pathway sequence. So the rough endoplasmic reticulum was the synthesis site, and see the rest of the pathway from one location to the next until the process has been completed. Okay, so again, you have there your pulses, which are exposed to the hot amino acids, and we have the unlabeled amino acids or the chases for the post-chase trials. So here you will see the representation of the autoradiography that reveals the sites of synthesis and subsequent transport of the secretory proteins. So figure A is the electron micrograph of a section of the pancreatic assigner cell that has been incubated for 3 minutes in the radioactive amino acids and then immediately fixed and prepared for autoradiography. So the black silver grains that appear in the emulsion following development are localized over the endoplasmic reticulum. So these are the silver grains, and they are found in the rough ER. So diagrams B to D is a sequence of the autoradiographs showing the movement of the labeled secretory proteins through a pancreatic assigner cell. So this is the red ones, okay? So the red ones there are actually the silver grains that you see here on DEM, okay? And these are, or this is how it actually, or they traveled from the different sites in the endoplasmic reticulum going out of the, or going out to the lumen, okay? So insights gained from the use of the green fluorescent protein. So if a while ago we discussed the use of the chase and the pulse autoradiography, here they actually use the green fluorescent proteins or the GFPs. So scientists can follow within their own eyes the dynamic movements of the specific proteins as they move with single living cell and do not have to kill cells. So the GFP is a small protein from the certain jellyfish that emits a green fluorescent light. And here you will see infected cells with vesicular stomatitis virus, strain in which a viral gene or the VSVG is fused to the GFP gene, and viruses useful since they can turn cells into factory of producing viral proteins. So as you see again, of course, with the use of these fluorescent dyes, you will be able to see how the movement of the proteins happen in the living cell. Okay? So in here, the figure here below with the viral genome shows you how, um, you know, these viral proteins, and of course with the addition of temperature, diffuses or fuses the vesicular viral gene. Okay? So how the changes happen. So insights gained from the biochemical analysis of subcellular fractions. So in this type of study naman, they used the technique of cell homogenization and organelle isolation techniques, which were pioneered by Albert Claude and Christian Deduve in the 1950s and 1960s. So homogenized cells form cytoplasmic membrane fragments, and the ends of which fused to form spherical vesicles around 100 nanometers in diameter. So vesicles formed from the different organelles have varied properties which allow their separation or cell fractionation from one another. So examples of the uses and findings are the following. Vesicles from different parts of the Golgi were found to have enzymes that add different sugars to the ends of a growing protein chains of glycoprotein or glycolipids, and identification of proteins in cell fractions taken to new level using sophisticated proteomic technology, and that wherein you can isolate organelle, extract and separate proteins, and then identify them by mass spectrometry. Okay, so usually guys, this cell fractionation, I think you have done this in maybe anatomy or physiology or chemistry, wherein you fractionate contents of the blood. Diba? With cellular subfractionations kasi, you filter, you separate the liquid forms to the more solid forms of a certain mixture, right? So that is also the same technique that this two scientists used for the subcellular fractionations. And then from there, from obtaining those cellular fractions, that's the only time that they dealt or studied each fraction individually and identified this um, or the components by the use of the mass spectrometry. Okay? So insights gained from the use of the cell-free systems. So isolated parts of the cell studied for their capabilities. So these cell-free systems provide information about the complex processes that were impossible to study using the intact cells. So George Palad and Philip Sikevitz et al. From the Rockefeller University in the 1960s studied properties of rough and microsomal fraction. So over the past few decades, cell-free systems have been used to identify the roles of many of the proteins involved in the membrane trafficking. And the examples below are, or the example would be the body of the liposomes. And then other insights gained from the study of mutants or mutations. So a mutant is an organism whose chromosomes contain one or more genes that encode abnormal proteins. So mutant gene products vary from the normal and they can cause characteristic deficiency in cell carrying the mutation which is analyzed. So one of the people who studied this one is Randy Sheckman et al. From the University of California in UC Berkeley. So he studied the genetic basis of secretion using yeast cells. Yeah. So what are the lessons that were learned from this technique? So first, dynamic activities of the endomembrane system are highly conserved, meaning when you say highly conserved, they are in, in a successive manner or in an orderly fashion. Okay? And processes are actually similar in all organisms like the yeast, the plant, insects, and human cells. And they are done with remarkably similar proteins, which are, or despite their structural diversity, these cells have underlying molecular similarities. Okay? So, for more information about this lecture, you can read CARP. Okay, so if you have questions about this lecture, just let me know, and I'll be more than willing to help you understand this one. Okay, so that actually ends our lesson for today. Thank you.